next speaker. And I'm very honored that she agreed to join us from US. Dr. Sridevi Devataj, she's Medical Director, Clinical Chemistry and Point of Care Technology, Texas Children's Hospital. And she's Professor of Pathology and Immunology, Baylor's College of Medicine. Actually, she hails from Chennai, India. She is a board certified clinical chemist and is presently medical director of the clinical laboratories at Texas Children's Hospital and professor with tenure at Baylor's College of Medicine. She has published more than 375 papers, 375, amazing, and has received many honors and awards from AACC, including the Grannies and Cooper and Zach Award. And her research is funded by NIH, and Dr. Devataj is also part of Global Lab Quality Initiative. And she is also actively involved with IFCC and inaugural speaker of ACC India section. And she was a mentor for ACC India section when we started that ACC India section. So we are really privileged to have you, Dr. Devataj, and she is in College of American Pathology's Technical Committee. And her topic for today is method validation. So Dr. Devataj, please.
Thank you very much. Especially with the pandemic, we realized very quickly how important it is for us to provide quality tests. And so how do we do that? By doing good method selection and validation. So first of all, as the medical director of a clinical lab, how do you actually choose which method you have to use? So first of all, if somebody comes, you have to establish the need for a test, right? And then once you establish that, yes, it needs to be offered locally, then you select the method, right? So how do you select a method? Is this possible to be done in my lab? Does it have to be sent out? Is the things that you have to think about. You define quality goals, and we'll talk about that later. You develop the methods. Most times, fortunately for us, we have manufacturers developing the method for us, and most often we are doing the evaluation or validation of these methods that have been developed to implement them in our clinical laboratories. And it's especially important when we have different laboratories having different instruments that this method validation is done properly. And then we have, before you go to routine analysis, you use QC practices and then result reports. So while the physician sees submission of specimen and the report of the result, these are all the steps that have to be taken for a result to be reported correctly. Okay, so the reasons for selecting a new method would be that, yes, somebody came, your GI physician came and said, hey, we have this new test. I want to offer calprotectin on all the patients that come in because I want to treat them effectively before they leave the hospital. So clinical need for a new analyte. And this new analyte should actually not just be another one that you can charge for, but it should be something that improves either diagnosis, treatment, or risk stratification, or provide better turnaround time. So we're a children's hospital. Instead of sending out a organic acid or amino acid analysis, if I had it in-house, then my NICU, my neonatologists are very happy with me because they can treat the patient effectively ASAP, right? So it should be an analyte that improves any one of these processes. It has to improve accuracy and or precision over existing methods. So you don't want to go back from a mass spec method to an RIA, but you want to go forward and use a better method, right? Then the other thing is reduce reagent and labor cost. And obviously, whenever you're moving a method from a manual method to an automated method, you're going to reduce reagent cost as well as labor cost. Now, labor cost in developed countries like the United States obviously is much more, and it's hard to get labor. So we're always looking to automate methods. And obviously, if you have a new analyzer or instrument, you want to validate these methods. So when it comes to method selection in the lab, you have to determine the analytical performance, how it does well in the lab, as well as the clinical performance. How does it improve patient care, right? So these are the two things that you have to keep in mind. The second one may not be very evident right away. And then depending on the test, you either do a method validation or you do a method verification. So so what is method validation and what is method verification? So the analytical verification is a process by which a lab determines. So here we have FDA approved tests and non-FDA approved tests, and you have probably NABL approved tests and not. So verification is a process where you make sure that whatever is written in the product insert that the manufacturer provides, you actually verify it in your lab and make sure that it actually performs to that criteria, right? Now, validation, what is the difference between validation and verification? The validation is usually for a test for which all of the characteristics have not been set forth by the manufacturer. And so the onus is on the laboratory to actually validate every single step of the method. So what are the different points that we need to take care of, okay? So here's your verification. That is for, I would say, most of the tests, because these are usually approved tests that are being done. Of course, there were a lot of COVID tests that were not approved and came off the market, for which you had to do a full validation, and it was not possible at the time. But let's not talk about that. For most other methods, if you're looking at verification, you do, this is a mnemonic, PARR. You look at precision, you look at accuracy, you look at reportable range, and you do the reference intervals. For the validation, you do precision, accuracy, reportable range, and reference interval. But in addition to this PARR, you also have to do analytical sensitivity or limit of detection, analytical specificity or interferences. You have to establish calibration and control procedures and other performance criteria, such as what's going to be talked about by the next speaker, which is like proficiency testing or interlab comparison. So the mnemonic for verification is PARR, if you want to remember that. And the mnemonic for the validation is the PARR plus the analytical sensitivity and the analytical specificity. Okay, when you have a goal to validate a method, so what do you need to do? So before you actually start on the validation, you need to figure out what is my acceptable performance for this test for precision, accuracy, sensitivity, and the measurement range. So if it's a test like triglycerides, your precision does not have to be 1 or 2%. If it's HbA1c, it has to be 5 or 6%. So you actually have to determine what the acceptable performance is. A lot of the times, these are also published already on websites that you can pull. The second is you assess the type of error and the magnitude of error. And this is usually from literature guidelines or professional judgment or physician input, and we'll go through examples. The third step, obviously, is finally, if the method performs to the specifications that you have generated, then you accept the method, or you say, no, I'm not going to go with this method because you don't want to compromise on quality. Okay, so the steps in the method validation are precision, accuracy, reportable range, reference intervals. We also do interference studies, calibration procedures, and finally, so these are all of the different steps that need to be taken care of when you're evaluating a method in the lab. Okay, so let's look first at precision. So the precision is basically the lab has to verify or establish analytical precision for each test using a sufficient number of characterized samples. So depending on what this is, you can do it by repeat measurements of samples at varying concentrations or activities within run and between run over a period of time. Okay, so what are the different types of precision that you have? You have the within run or intra-assay precision. So which means that within each run, you do 20 samples, 40 samples, whatever it is in one run, and that's your intra-assay precision. Then you have your between run precision, which is you can do it 2 times in a day, 3 times in a day, 10 samples, 20 samples, and you have your between run precision. And then you have your day-to-day precision, which is your total precision. So why do we have to do these different kinds of precision? And we'll go through that. So the CLSI has guidelines for these precision studies, and they say for within run precision, 20 consecutive replicates in a single run is good. For the total, so total would mean between run and between day, right? So you can either do 2 runs a day for 20 days with 2 replicates per run, or if it's just a verification and you want to make sure that the precision is fine, then you do 5 samples over the range of the analytical measurement range, and you do it times 5, and that's your verification done. So how do you analyze your data? You calculate your mean ST and CV, and obviously you have already determined what your percent CV should be, and you do an analysis of variance. So random error. What is random analytical error? Anything that is caused outside because of environmental condition, either the instability of the instrument, or if you have variations in temperature, and that can happen a lot in labs here as well as in India. I'm in Houston, so you have very varied temperatures again, and you can have variations in humidity, variations in the reagents and calibrators. The previous speaker just talked about shortages of reagents and calibrators and having to cross-validate quickly across methods as reagents were becoming unavailable. So, right, variations in the reagents you have to check. Variation in handling techniques, such as how good is somebody at pipetting? Are they mixing it properly? Are they centrifuging for 4 minutes? Or too many samples and they're centrifuging for 2 minutes? What is happening, right? Is there a variability in these operators? So this is all referred to as the random error, and you can pick up the random error by doing all the 3: the within run, the within day, which is between run, and the between day. Okay, so if you go through each one, what does it pick up? The within run component of variation will pick up differences in sampling, precision, if there's really short-term variations in temperature, and the stability of the instrument. If sample 1 were to sample 20, is it running properly, right? Within day or between run, that is caused by instability of the calibration curve, which means that you then have to recalibrate. Hopefully, we want a method that is stable, that you don't have to recalibrate more than once a day, right? And then between run, long-term variations in the instrument, small changes in the calibrator and reagents you can pick up, and changes in the condition of the lab during the day. And obviously, somebody that's coming at 8 a.m. versus 4 p.m., how good are they, especially if it is a manual technique that's being run. Between day, again, it's caused by daily variations in the instrument and changes in calibrators and reagents, especially if you're opening new vials each day for a high volume test. Changes in staff from day to day. Hopefully, they're all well trained and they're following the exact same SOP, and so your between day variation is less. So all of these cause changes to the precision. Okay, now that you have established your precision, you have a great intra-assay and inter-assay precision. So then you go to the accuracy. So how do you establish accuracy, right? Or comparison of methods. So this is our famous or infamous bullseye diagram. So this is something that has really bad accuracy as well as precision, because this is your target and nothing is in your target. This excellent precision, but as you can see, there's a huge error because it's not at the target, it's much higher out there. So this is again high accuracy, but the precision is not great. You have 3 of them that are within the bullseye and then the rest outside. And obviously you want something, all your methods to be this way with really high accuracy and really high precision. So for the accuracy studies, what are the guidelines? So you select a comparative method. So select a comparative method that is a gold standard method. If the gold standard method is not available, try to do samples with a lab that does have a gold standard method or has been previously validated to a gold standard method. And it's important that you familiarize yourself to the instrument that the method is being done on before you start the accuracy studies. The method comparison, the CLSI again recommends that you analyze more than 40 specimens by the test and the reference method over 5 days. You estimate bias, regression characteristics and plot and compare the results to judge the acceptability. So it's depending on what kind of method it is, whether it's a quantitative method or a qualitative method, you're going to have different goals for your accuracy studies. So again, if it's an LDT, what an LDT is a lab developed test, and we have had several of the lab developed tests that have occurred here because of the pandemic. And so you have to validate the analytic accuracy with an appropriate number of samples. For quantitative tests, the minimum is 20 samples across the analytical measurement range. For qualitative tests, also you do 20 samples, but divide it into positive samples and negative samples and check for low positive samples. So at the concentration near the cutoff where it's going to be positive. So you don't want to use an equivocal sample, which is neither positive nor negative. So use negative samples, positive samples, and something close to the cutoff range so that you are actually verifying the qualitative test, right? The nature of the test. So if you're not able to get 20 samples and it's a, it's a analyte, which is kind of rare, then the lab director must record the criteria used to determine the appropriateness of the sample size. Okay, and this we all know how to do. We use different programs. Some people use Excel, some people use EP Evaluator, but basically you do the X is the reference method, Y is the result of the new method, and you can plot the two, and you want to determine the correlation coefficient. So R is the statistical term that is used, and ideally if there's a line of identity between the test method and reference method, you want the R to be 1, okay? Okay, so then what do? You generate the linear best fit line. And I'm giving you an example here, just of a general one in the lab. Here's the test method, the comparative method, and you're looking at, this is in millimoles, so that's why it's less. It's not milligrams per deciliter. So you look at it over the analytical measurement range. You have done at least 20 samples. You have a slope of 1 and an R of 0.99. So there's your method. So then you establish what is your systemic error, especially if you have a analyte for which you need medical decision points, like BNP, like troponin. So you have to actually determine the systemic error at your medical decision point, because you're saying somebody needs to be treated for an MI because your troponin is high and is rising. So then you want to make sure that at that medical decision level, that your systematic error is as low as possible, and you want to compare the result tracking over time. So here is the systemic error. So this is the line of ideal line. There's your reference method and your new method, and there's the ideal line where Y equals X, right? Now there's something called a constant error. Here there's no error, obviously. Sometimes it's a constant error, so it's always 10% over the line of identity. So you know that using my method, my reference intervals are going to be different. My decision cutoff point is going to be different compared to the gold standard method. However, it's much harder when it's not a constant error, but a proportional error. And then you know that over a certain limit, you're going to have a wide variability in your test, right? So these are things that you need to think about. So the total error, again, this is the true value and this is the observed value. The total error equal to the random error plus the systemic error. And you have to establish these criteria if you already don't have it established, for example, in the Westcott website, or your CLSI website, or your CAP website. You may have local total analytical errors published as well. And you do your Bland-Altman plot to show differences. It's easy to say whether it's a random error, whether it's a proportional error, or a constant error, and you want your all the points to be stacked around this line at 0. Okay, so you did your precision, you did your accuracy, now you have to do your reportable range or linearity, right? So what is the reportable range? So the reportable range is also referred to as the analytical measurement range. And these are basically you have the true concentration of the analyte, and you're checking to see if your method actually performs according to the published range of that particular analyte. So this is the range where the method can directly measure without any modification, no concentration, no dilution, no other treatment that are not part of the usual assay process. Now there's something else called maximum dilution or concentration, and this was referred to as a clinical reportable range. Now this is very important because sometimes you go much higher over the analytical measurement range. For example, if you want to measure an AFP for HCC, right, hepatocellular carcinoma, or you want to measure the HCG in pregnancy, it can go up to 100,000. So you have to actually verify your clinical reportable range. Say your analytical measurement range was up to 5,000, but you want to go up to 500,000, then you need to do a dilution of your 5,000 and make sure that a 1 to 100 comes down to 50 reliably, right? So then you have verified your dilution process. So you can extend your AMR by 100. So your 5,000 can then become 500,000, right? So this is how you do your AMR versus your CRR or maximum dilution. And again, if it's value of higher than AMR, you report as greater than. If you have established a CRR or MDC, then you dilute and auto dilute based on your instrument. Okay, so the reportable range, the ways to generate a sample for the linearity study is you either spike a low sample with a known amount of analyte, or you keep diluting the high sample with a blank so that you get a curve, or you can mix a high and a low sample. Most often you have linearity material that's available from, say, Biorad, the many companies, I'm sure even in India, that offer standard reference materials. And you also have commercial linearity products that you can use. So what does the CLSI recommend? That the testing sequence should be random, and it's best to have at least 5 concentration of the analyte over the analytical measurement range. And you do want to verify the lower and the higher, so that you need to be at least within your total allowable error of the low and the high. For example, if you say your analytical measurement for glucose is anywhere from 25 to 700, you need to have a sample closer to 600 or 700. You need to have a sample closer to that 45 so that you can pick up hypoglycemia well, you can pick up hyperglycemia well as well, okay? So that's what you do for your reportable range studies. So you did your precision, you did your accuracy, you did your reportable range, and then lastly, if you're just verifying, you need to do your reference intervals. Okay, so for the reference intervals, these are difficult, okay? So especially in certain situations where you're doing tests for specialized populations, it could be HIV-related tests, it could be an OB, obstetric population, it could be a pediatric population where you don't really get blood unless a baby is sick, right? So it's very difficult to establish, for example, neonatal reference ranges, right? It's probably a little easier in places like Asian countries where the population is higher. Very, very difficult to do it in other places. So what we do is either verify existing intervals, or we do transference of reference intervals. How do we do that? So you can verify the existing intervals. So the manufacturer has published an approximate reference interval, or you look in the literature and you know that your reference interval for, for whatever analyte, AST or ALT, SGOT, SGPT is certain thing, or you construct new reference intervals because it's different. So for example, this 5 to 15 micromolar for homocysteine is a great reference interval. Is that really a good reference interval in the Indian population? Do you need to construct a new one then? So these are things that you need to think about. So the good practice is to verify that the reference interval is appropriate for the patient population that is being studied and for each specimen type, okay? It may be sometimes that your serum reference values are different from that in urine, in CSF, and you have to, establish that. Now, transference of a reference interval, like I told you, is acceptable if the test subject and population and methodology are same. And how you can verify it is by testing at least 20 samples. And if less than 2 are outside the limit, then you accept that and you transfer the reference interval. So we do a lot of transference, for example, in the pediatric population. So CALIPER is a study that is a huge pediatric reference study that was done in Canada from neonates till age 18. So a lot of times, and they've done it on different instruments as well. And so we're able to transfer those reference intervals by doing just 20 samples here locally. So 95% is usually the values that you establish for the study population. And for that, the CLSI guideline says for adults, new method, you do 120. Most often you do 40. And again, in specialized populations, you can do 20. Okay, so what do you need to do? You establish all of the selection criteria for the individuals, your confidence limits, whether it's 95%, 90%, and then use a histogram to evaluate the distribution. And obviously, you can only use it if you do, if there's a bell-shaped curve or a Gaussian distribution, and you can leave out the 5% on this side and that side, and you can get your reference interval, right? Now, if it is non-parametric, also there are other ways to do it using certain other criteria, like your median and interquartile range. Okay, so you've actually verified your test now because you did the PARR, right? You did the precision, you did accuracy, you did a reportable range, you do reference interval. Now, if you have to do the entire method validation and not verification, then what do? You have to look at analytical sensitivity. So there's different terminologies. So I'm going to talk about this. So analytical sensitivity means you do limit of absence or limit of blank. And that is the lowest concentration of the analyte that the method can differentiate from 0. So if you have a 0 and you can measure a 0.1, that's your limit of the blank always reads 0.1. This is usually done for things like CRP. You run your specimen diluent, it already gives you a range of 0.5. So your limit of blank for CRP would be 0.5. Then you have the limit of detection, which is the minimum concentration that you can detect appropriately. And the function, sometimes you also have limit of quantification, which is different from the limit of detection. So let me go and show you what this is. So if you have here, this is your limit of blank. If you run your solvent, that's where you get your limit of blank, right? And you can do this 10 times, 20 times, and establish your limit of blank. Your limit of detection, it's always greater than your limit of blank. And you can actually detect it from your method, but your CV is very high. And so you want to go to a little higher limit of quantitation where your total error is very less, okay? So the limit of quantification is the lowest concentration that can be quantified reliably. This is especially true for your HPLC methods, for your mass spec methods, and for methods that are mostly manual. You usually have the limit of quantitation. And anything that's below the limit of quantification, you report as less than LOQ. And that's exactly what I said. And this is what we're talking about, observed by the predicted. You want to measure something that's over here. So 0.5% is your CV that you have accepted. So anything that's over your 0.5 is something that you will use as your limit of quantification. And this is just showing in graphical form. This is the limit of absence or blank. This is the limit of detection. You can pick it up, but it's not still grade CV. And your functional sensitivity is nothing but your limit of quantitation. Okay, so you did your sensitivity. Of course, you have to do specificity or interference studies, right? So what is analytical specificity? So you have to do, you have to look at methods that are possibly interfering or cross-reacting to the method of, to the analyte that you're measuring. So let's go and see what are these. So the CLSI again has guidelines for these, and they say do a paired difference study. So you do one with the potential interferent, one without, you calculate the differences, and you can do this with patient samples, and you do the bias, make sure it's within your total allowable error. So for the most classical interferences that we normally find are lipemia. So if you have a sample postprandial, somebody comes in and they've eaten a fatty diet, you want to make sure that the lipid doesn't affect the analyte that you're measuring. Hemolysis, this is very common in pediatrics because, you know, of not being able to draw the blood properly, especially if you're drawing capillary draws. So hemolysis is something that you have to measure. And obviously, only analytes that are within the RBC are ones that you're most concerned about with regards to hemolysis, like potassium, like CK, like LDH, right? Like AST, those are the ones, ammonia, these are the ones that you look at hemolysis. Bilirubin, icteric samples, again, you look for the interference, and most often it's an interference because of the spectrophotometry, and that anything that absorbs close to bilirubin in the 400 range, then you want to check that with your analyte to make sure it doesn't interfere. Less common substances that can interfere are drugs. You can also have immunoassay interferences like heterophile antibodies. You can have anti-mouse antibodies, anti-rabbit antibodies, and you can use a pre-immune serum to test for those. Rheumatoid factor and immunoglobulins, if you have high concentrations of immunoglobulins, especially in immunoassays like prolactin, TSH, these are things that you want to test for interferences with these factors. And another common one that has been published a lot these days is also biotin. Biotin again interferes with a lot of and it's in a lot of antioxidants, it's in a lot of natural supplements, and it can affect a lot of these immunoassays. Why does it affect the immunoassays? Because you have a biotin-avidin reaction in your immunoassay. And if you have external biotin, that can compete with the biotin-avidin interaction that is necessary for measuring your metabolism, right? I'm going to go through that. And yeah, we talked about immunoglobulins. You can have immune complexes. Classically, you know, things that you can have macroprolactin, macro CK, those are things, macro AST. In fact, you can have non-specific binding because of immunoglobulins, non-specifically, you know, binding to these analytes, and you need to check this interference in immunoassays. Another one that's common now in immunoassays and electrophoresis, especially, is the fact that you have all of these monoclonal antibody therapies, which are fantastic, but can cause an interference, right? So in fact, in one of the earliest COVID tests that was in Australia, they kept saying that a lot of people, you know, had positives and they were actually not having the infection, and it was because of interference with one of the medications that they were taking, right? So it's very important for us to establish which interferes with your analyte. So how you would do the interference experiment Again, pairs of test samples, you measure the analyte A in both samples, you calculate the difference, and you obviously want a 0% bias, but you can also establish a bias. So there you verify your analytical specificity. And if there are certain things that interfere with the test, put a disclaimer along with your test result saying X can interfere with this result, right? So that the physician is aware. So once you've done all of this, you obviously have to document for, especially if you're looking at certification and your inspectors are coming to look at these, you want to look at how you document an approval of validation. So how do you actually do this? So for every method that you validate, there needs to be a validation or verification protocol and a summary. So it should include a written assessment of each component of the study. If it's a verification, it's PARR. If it's a validation, it's PARR plus analytical sensitivity and specificity. And you have to have a statement about the acceptability of data, and then a signed approval statement and investigation of any discordant results. So this is a template that is available in the College of American Pathologists. This is the analytical verification template. So you can tell which test on which method, your summary of your accuracy, precision, reportable range, and reference interval studies, and this statement saying that you as a director have reviewed all of these for this method, and you find it acceptable for patient testing. So before the testing goes live, you approve it and sign it. And so you've blessed the method. So you can establish whether your total allowable error is 15% or not 10%. Obviously, you don't want to fix the data. You want to do that before you actually do the verification and make sure that it passes. And this is for the validation. You have an analytical validation template with all of the things that I talked to you about, the PARR plus the analytical sensitivity and the specificity. And again, you have the statement, you approve it, and you sign it, and that's it, you're done. Now, the other considerations is, I do want to talk about medical decision, simple things like even glucose. If you want it to be a less than 100 or a greater than 125 for impaired fasting glucose or for diabetes, you want to make sure that at those levels that the CV is excellent and that it compares to a gold standard method, okay, for troponin as well. We're almost there, 2 more slides. And so the other thing is when you're moving methods, when you're moving instruments, you need to do a little verification study to make sure that your instrument, even when you're moved to a different location where there's different temperature, humidity, etc., different people running it, that you validate that. And any test modifications, you have to validate, okay? If you're doing a urine and a serum assay, if you don't validate it, it is not acceptable. If you do it in a non-approved patient population, it's approved in adults, but not in children, not acceptable unless you do a validation. Qualitative versus quantitative. Oh yeah, for the antibody test, I get numbers. I want to give the numbers. That's a no-no, unless you validate it as a quantitative. The incubation times and temperatures, if you're changing, especially for a PCR, obviously you have to validate that. Any dilution other than what manufacturer recommends has to be validated. Any eliminating a procedural step in a manual assay, that step might be very critical. So you have to validate that. The intended use has to be appropriate, and specimen stability, obviously, because there are certain tests like steroids that are not very stable. There are some that have to be refrigerated, otherwise it's not stable. Any test modification that's done to the manufacturer's guidelines have to be validated. Okay, so again, I want to end with this slide where talking about, this is the gist of what we talked about today for the method validation, your verification parameters and your validation parameters. Obviously, PT and training we didn't talk about, but those are important things while you establish a method and you report a result. And every single sample, like the previous speaker mentioned, is not just a sample, it's a human being at the end of the sample. And as long as you're always aware of that, you're going to produce quality results. So I'll stop with this, and any questions, please, please feel free to ask.
Thank you, Dr. Devataj, for elaborating validation. I think this is one of the most challenging in our lab life, doing the proper method validation and also following the verification protocol. So we are privileged to have you, and here I can see there are many attendees and delegates from even the neighboring countries. So one is like Dr. Saifuddin Khaled from Bangladesh. So he asked about harmonization, how harmonization can be done. How can clinicians or users complain can be answered if they do the test in different lab with different platforms and experience totally different diagnostic cutoff.
Right.
So this is actually, yeah, India, Bangladesh, and neighboring countries. We see like, one patient goes to 2-3 different labs during the same time because we are not very much insurance driven. So this is the challenge.
Well, let me tell you, it's a challenge here as well. Even though we have everything in the electronic medical record, that's even more of an issue because everybody everywhere, it's called Epic everywhere, right? So everybody can see it. So it's important that you do standardization. How do you harmonize, right? If you have international standards, you can harmonize to those international standards. So IFCC has a whole list of international standards. The WHO has a set of international standards. And obviously, if your manufacturer is doing the work of standardizing it to the WHO or the IFCC standard, nothing like that, right? But obviously, that is few and far between. And that's why the onus is on us to actually establish these harmonization rules. And a lot of it is driven by local guidelines and local standards. So even we have a organization now, which is called the True Lab. And basically what we are doing here in the United States, at least, and Canada, and some of the South American countries as well, is to actually look at, how we harmonize all of the different tests based on line codes, right? So it is a challenge. I agree with that. But as long as you are able to report, so for example, we all classical, right? PTH. So you have a patient in the hospital with intraoperative PTH. You look at the test, it's a 250, come down to 50, and you send the patient home. You want to follow up, they go to a Quest lab, which is close to their house, and it's a 250 again. And you're like, what is this? I did surgery, I took out the parathyroid, where's this coming from? But then it's coming from the fact that the Quest assay is 2.5 times your assay. So it's very important also in the report to mention which method you're using, so that for a clinician that's looking at it, they're looking at, okay, PTH Abbott, PTH Beckman, and they know that it's different. So any clues that we can give, like most often a glucose is a glucose is a glucose everywhere. That's pretty well standardized, right? But not a TSH, not a PTH. So these are things that we need to actually point to the physician to actually pay attention to. I hope I answered the question.
Thank you. Thank you so much for this excellent talk.